today's talk is really focusing on the next step, development. That's how can we efficiently deliver an API to its target without any adverse effects. And so for that, we're combining our expertise in materials science with our expertise in pharmaceutical scientists in order to start building out our platform to support this question. So I've divided this talk into three main sections. First, I want to give a brief overview of molecular dynamics and then jump into our two main topics, which are cosolvent solubility and amorphous solid dispersions. Before jumping into the physics-based models, I did want to start with actually our machine learned model. This allows for really fast computation and estimations of solubility, and hopefully you'll see why I positioned it here. But we've trained a large data set based on some literature on over 28,000 total experimental data points that cover 128 different drugs and 44 solvents. And you can see that from the test set, the set of data that's withheld from the model while it's being trained is relatively well predicted. So you have a test R-squared in 0.93. So these tests on the unseen data are giving good results, even for binary mixtures and for temperature dependence, both really instrumental to this cosolvent selection process. In the bottom of a lot of these slides, I want to point out while I'm here, I have this kind of little clock icon, and that's meant to kind of give an idea of how long it takes to run these. So in order to evaluate one cosolvent selection or even an array of them, it really only takes a second or less. It's almost instantaneous in order to get an answer. And so that's really where the power of something like a trained ML model can come in, as well as other types. How can we handle APIs and solvent combinations, especially as we are generating more and more unique APIs outside of the chemical space of the ML model? And that's really where we think physics-based modeling has a role. So one of the first tools we developed in this space was the FEP plus solubility workflow. So the simulation technique for computing specifically aqueous solubility. And so it's an intractable problem to actually directly measure delta G solubility. So instead we break it down into a thermodynamic cycle and look at two separate legs, this delta G sublimation and delta G hydration. Both of those we can get directly through simulation. So what I wanted to focus about more in this section is actually just this one solvation-free energy leg. So instead of just hydration, where we're looking at free energy into water, we're looking at solvation by any arbitrary solvent. And so we can actually relate the difference in the free energies, which we attain directly from simulations, to the solubility ratio, which we can directly obtain from experiment. So the solubility ratio is really saying how much more soluble is solvent 1 than solvent 2. So we tested this in pure solvents first, and we find that it accurately predicts trends in solubility. So what's shown here are 3 different API molecules and 4 different solvents. And the trends are quite good. The R-squareds are pretty high. And we can actually, if we know the solubility in one solvent, which is not shown here with ethanol, then we can predict the solubility in any solvent. We wanted to see how this worked with cosolvents. We know that once you have 2 solvents in there, the solubility is going to change. And in some cases, there's some synergistic effects between 2 different solvents. So shown here is just the solubility ratio, all normalized relative to pure saccharin or methanol and pure saccharin and pure methanol. And then we see how the solubility changes relative to that as we add anisole. And so you can see experimentally, you generally have an increase in the solubility as you add anisole until it's pure anisole. So there's this synergistic interaction happening. And our FEP plus actually gets the shape and sign of that curve right, including identifying the correct optimum value. Whereas a semi-empirical method like Cosmo, which works great in pure solvents, might have some difficulties. And in this case, it actually reports back a monotonic trend, which is generally in the wrong sign in all these cases. So this is an example of where incorporating the many body effects that are in a physics-based simulations actually benefits our predictions. So these have been relatively simple solvents. We wanted to continue to push these boundaries and look at more complex solvents. And so moving from these small ones up into the oligomer space. So PEG 400, mixtures of PEG 400 and water was one of the next things we studied. For this particular case study, I was interested in knowing what's the partition coefficient between polyethylene and water. So what's the solubility ratio as another way to put it. And in this case, we do see some pretty good predictions where you have very good correlation. Once you have a polymer, we need a way to build an actual simulation cell, which we'll use to run our final MD simulations. Our disordered system builder actually has some really powerful functionality in order to build polymer chains in a way that's initially very entangled, which is where you'd expect. So you can see you're really building a system that kind of matches the vision that we have of what an ASD looks like on a molecular level. And that's before we do any equilibration. We start with 350,000 atoms, which is quite large, and then we slowly evaporate them until we're left with only 20,000 total atoms in the cell. And so it's really interesting to watch this because we can see how the polymer changes of H3MCAS are move and orient with respect to the type of solvent. Maybe we can look into things like radius regeneration to see how that changes with different solvents. Another type of coarse-grained procedure that we use is dissipative particle dynamics. We can use, for this particular system, we're able to map it in a way that we can make even larger particles and represent 20,000 beads, and use 20,000 beads to represent 380,000 atoms and run at approximately 10,000 nanoseconds per day. So you see how that, you can hopefully graphically now see how we can accelerate our simulations. This is some work that we did in collaboration with AbbVie in order to create a coarse-grained model of ibuprofen and capovidone. So what we're showing here is, ibuprofen converted to a coarse grain representation. The spheres overlapping the atoms in the all atom ibuprofen structure are used to define the centers of the beads in the coarse grain representation. And so we actually are able to map 33 total atoms down to just 2 particles. Copovidone, we do something similar for just the monomers, and then we can actually build monomers. We can build polymers from these monomers, and on average, you're reducing it from around 142 atoms to 10 total particles. So a significant reduction in system size, which will come with a commensurate increase in simulation speed. Now, you might be asking, how do I choose where to place these spheres? How do I give these parameters so it acts like a real molecule? Because in an atomistic simulation, it's pretty straightforward. We know what the atoms do. So for that, we've actually heavily invested in an automated procedure for parameterizing these models. Right now, it works for DPD, and it's on our roadmap to have it working with Martini as well.